All right, guys, today we are going to look at this 8.2 active reader. It's worth 10 points. And you've done these before. You go through, you read the paragraph, and you answer the questions that follow. So I did this one a little bit different. I have the reading as question number 1, and then the actual questions are in question number 2. So let's go through and just read our paragraphs here first. It's all about mitosis, so these are things we've already covered in our notes. Mitosis is a continuous process that is divided into 4 phases: prophase, metaphase, anaphase, and telophase. Prophase is the first phase of mitosis. Prophase begins with the shortening and tight coiling of DNA into rod-shaped chromosomes that can be seen with a light microscope. During the S phase, each chromosome is copied. The 2 copies of each chromosome, called chromatids, stay connected to one another by the centromere. At this time, the nucleolus and the nuclear membrane break down and disappear. 2 pairs of dark spots called centrosomes appear next to the disappearing nucleus. The centrosomes move toward opposite poles of the cell, and spindle fibers radiate from the centrosomes in preparation for mitosis. Metaphase is the second phase of mitosis. During metaphase, kinetochore fibers move the chromosomes to the center of the dividing cell. During anaphase, the chromatids of each chromosome separate at the centromere and slowly move toward opposite poles of the dividing cell. After the chromatids separate, they are considered to be individual chromosomes. Telophase is the fourth phase of mitosis. After the chromosomes reach opposite ends of the cell, the spindle fibers disassemble and the chromosomes return to a less tightly coiled chromatin state. A nuclear envelope forms around each set of chromosomes, and a nucleolus forms in each of the newly forming cells. Use the rating above to answer the questions that follow. Did you read through everything? You get a point just for answering this question. You should answer yes, because if you're watching this video, I just read you the entire thing. If you're doing it on your own, hopefully you read through it. Describe which state of mitosis the following events occur in. So for each one, you have the same 4 answers: prophase, metaphase, anaphase, and telophase. Chromatids of each chromosome separate at the centromere. So let's see, they separate. During anaphase, the chromatids of each chromosome separate at the centromere. So for this first one here, I'm going to go with anaphase. Copied DNA coils into chromosomes. Let's scroll back up here. Let's see, prophase, tightening and coiling. During the S phase, they're going to be copied. So we're going to go with prophase. Now, whenever we talked about this stuff before, we know that the coiling and everything actually happens, it starts to happen in interphase. So it's not exactly accurate with what they have written here. But anyway, spindle fibers disappear. So in prophase, they're starting to appear. They're radiating from the centrosomes. That means at the end of it, the spindle fibers are going to disassemble. So disassembling means that they're going to be disappearing. So we're going to go with telophase. Spindle fibers move chromosomes to the cell's center. So they're moving to the middle. Middle is going to be metaphase. Centrosomes appear next to the disappearing nucleus. That's going to be prophase. The nucleus disappears at the beginning. Nuclear envelope forms in each newly formed cells. So since we have newly formed cells, that's telling me it's telophase. Chromatids move toward opposite poles, so they're moving apart. Apart is anaphase. Chromosomes line up in the middle of the cell. Middle is metaphase. And a cleavage furrow or cell plate begin to occur is going to be in telophase. All right, guys, that's it for this active reader, and I will see you back here shortly.